Next in the cellular reproduction segment, I want to discuss basically the cell cycle. Again, give a brief overview of what the cell cycle is, the events that occur during the cell cycle that basically dictate the activities of the cell itself. Right. So basically, what the cell cycle is, it's a series of events that occur, obviously in a cyclical fashion. That basically, it's basically a summary of cellular activity. All right. And the cell cycle basically can be split up into four separate phases or four separate parts where there are different types and amounts of activity that are occurring. All right. So basically when the cell starts to go through this cell cycle, it's getting ready to divide. But cells don't just divide right away. Cells need to prepare for cell division, for the process of mitosis. All right, and that's what these first three phases of the cell cycle are all about. Basically, if you want to look at the labels on here, you can see that there's what's called G1, S, and G2, and then there's the M phase. That's what we think of as actual mitosis. All right. Like I said, the other three phases of the cycle are preparatory phases for the big game, for the splitting of the cell itself. All right. Now, the two-letter G stand for gap, and S stands for synthesis. All right. Now, one thing to keep in mind, like I said, this is a general overview of this. This is a lot more complex and in-depth than how I'm going to explain it. But like I said, later on I'll make some more videos about these individual parts of the cycle. Now, basically, if we kind of break this down step by step, talk about the different phases, like I said, just keep in mind all of this is leading up to mitosis itself. So, if we want to take a look at the cell cycle, all right, we can see that there's mitosis, cell division, and the other parts that lead up to it. Now, these first three stages are what we call interphase. All right, interphase. G1, S, and G2 are all basically, think of that as just regular activity of the cell and preparation for division. All right, and let's say for the sake of discussion that it takes 24 hours, that the cell has a 24-hour cycle for this whole process. It would spend on average about 23 hours out of the day in interphase. All right, preparing to divide, going through all the different steps of preparing to divide. And then it would spend, give or take, an hour actually undergoing the process of splitting into, identical genetic copies of itself, the daughter cells. All right, but bear in mind this isn't the same for all cells. There are certain cells that would spend maybe half of the amount of time in a day undergoing this. But just keep in mind that that the bulk of that time is spent in interphase. So if the cell cycle is only 12 hours for a certain type of cell, it's still going to spend 11 of those hours, give or take, in interphase, and one of those hours undergoing mitosis and division. All right, but like I said, those are just general numbers. There's a lot of variability, and we're made up of a very diverse group of cells. But that's kind of a concept I want you to keep in mind. All right. Now, when we take a look at the various phases of interphase. The two gap phases, these are all relatively easy to understand, but the two gap phases are realistically, they're just kind of, I don't want to say stall tactics, they're just kind of slowing things down, okay? Because in order for cells to grow and divide, there's a couple factors that need to be in place. One of them is, being cells need to be big enough. Cells need to have the right amount of nourishment and energy to undergo this process. Cells need to be exposed to the right chemical factors from the external environment. All right, so basically G1 and G2, that's really all they're doing. They're basically just delaying they're stalling the process to ensure that the cell is fully prepared to divide. All right, so basically what's going on, especially during G1, the first gap phase is just cellular growth. The cell is enlarging in size. Okay, it's increasing its surface area. It's making more organelles. It's increasing everything about itself. All right, along with just fairly normal activity that's going on as well. But another thing about this is that this is a very critical regulatory phase of the cell cycle. All right, in the previous video we discussed that one of the biggest problems that you see when cellular reproduction goes wrong is cancer, when cells grow and divide out of control. All right, but luckily cells have built-in mechanisms to help basically figure out if there are any problems during this process. All right, there are also external controls to this as well, but we'll focus on the internal mechanisms. Now, basically, what happens if, let's say, I don't know whether the environment is not conducive to the cell dividing. Maybe there's damage to the cell, whatever it may be. All right, that cell cannot divide. We can't afford to have that cell making a copy of itself if the potential is there to make a bad copy. So basically, what's going to happen then is the cell is going to put itself in what's called the G0 phase, gap 0. All right, and basically in gap 0, there's really nothing going. Think of it as a stage of hibernation for the cell. All right, so the cell is basically going to sit there fairly inactive until it can repair the problems. Whether it is damage to DNA, damage to an organelle, whatever it may be, the cell has to fix the problem before it can continue. All right, and there are different types of intrinsic regulatory mechanisms inside the cell. That we use all the time. Cyclin-dependent kinases. That's what CDKs are. And again, I'm just going to introduce these. I'm not going to go in depth. But this is a class of protein kinases within the cell itself that kind of go through their own cycle of regulating the cell, and they act as triggers to move the cell through these different phases. All right, and if something goes wrong and these triggers are unable to get the cell through, then it goes into G0. All right, there's also what are called maturation promotion factors, MFPs, that, again, very similarly act as triggers that, or switches that move the cell from phase to phase. If something is wrong with the cell, then we're unable to flip that switch, then we go into G0. All right, there's also, this is a very commonly studied gene, the P53 gene, making, the protein, P53 protein. All right, same thing. P53 also shuts down the cell if something is wrong. Okay, and this is, these are good because, like I said, we need these switches to turn the cell off, because if we make bad copies of itself, the chance of, forming neoplasms, new tissue growth, basically cancerous tissues, are high. So we need to make sure we suppress bad activity of the cell. And, that's what they look for. And one of the things they look for when they want to biopsy and test cancers, they want to see, are there mutations in these regulatory genes, in these regulatory proteins, all right? And if there are, there's a good, that's probably what threw the cell out of its normal array of activity. All right, and most cells in the body are in a perpetual state of G0. All right, basically they're just there, they're normally functioning. All right, but when the time is right, when we need a cell to divide, that's when we get into these, these G1 states. All right, the G1, S, G2, and the mitotic phase. All right, there are some cells, like I mentioned earlier, muscle cells and neurons. They're permanently in a state of G0. You can say that they're differentiated into this gap 0 phase. They're specialized in this. They lost their genes to induce and regulate division are gone. They're permanently shut off. Okay, so some cells in the body never leave G0. All right, gap 0 can last as long as a few minutes to a few days to years. It just depends on the extent of what's gone wrong with the cell and how long it's going to take for us to fix it, if we are going to be able to do that. Okay, so that's basically the gap 1 phase. Like I said, when we basically stimulate a lot of cellular growth is taking place along with the regular activity of the cell. And like I said, this is a very critical regulatory phase. And basically, if we pass through all the checkpoints of this, if everything is good, through G1, then basically these switches allow us to move into the S phase of the cell cycle. All right, and S stands for synthesis. All right, and really the main thing that's going on here is DNA replication. You have to remember that when we divide and we make genetic copies of daughter cells, these daughter cells are taking basically genetic information from the mother cell. All right, so as a result, this cell needs to replicate all of its genetic material, needs to replicate all of its DNA. All right, so we can ensure that we have exact identical copies. All right, and this is by far the longest part of interphase. This takes up generally, at the minimum, on average, about half of the overall time frame of interphase. All right, it takes a while to replicate the entire genome of that cell, and that's all that's going on there. And then then we'll transition into gap 2. And again, all it is just a stall tactic to ensure that the cell is growing to a good enough size and that everything is in place to allow the cell to divide itself. All right. And then, as you can see, these red lines represent switches or checkpoints. All right. So basically, if everything is in check and everything is good, then these switches will allow the cell to transition from phase to phase, throughout the cell cycle. So if everything is good throughout G1, S, and G2, then we'll be able to transition into the M phase, mitosis. All right, and I'll do a video more in detail of mitosis. But again, mitosis is basically the division of the cell. All right, the division of the nucleus and the transfer of the genetic material from that mother cell into the two daughter cells. All right, and the physical splitting. All right, and then we start all over. Then these cells then we have normal functioning daughter cells. They may just go right back to G0. They may have to divide. It depends on the situation that we're in. All right, but in a nutshell, that is the cell cycle and some of the activities that go on during the cell cycle. Like I said, all this is just preparation for the big show, for the actual division of the cell itself into the two daughter cells. Right, and again, remember these phases, G1, S, and G2. Like I said, there's a lot more going on here, more in depth than what I mentioned. But like I said, if you have a general understanding of this, you'll have a good grasp on understanding. Okay, we make it all the way to the M phase. Now we can undergo mitosis, and you can understand what goes wrong. What can potentially go wrong here as well to cause cancers and other problems.